All right, welcome back. So now we're going to talk a little bit more about action potentials and how they propagate. So the difference between unmyelinated versus myelinated axons. All right, so I'm going to hide me here. All right, so when we talk about an unmyelinated axon, take a look at this. We have all those, the same stuff's happening. Sodium channels are opening, potassium, voltage-gated potassium channels are opening, all that kind of stuff is happening. Okay, so here in the left-hand side here, what's happening is that we have the opening of sodium channels, but you have to have a sodium channel open and close and potassium channels open and close on every single part of the cell membrane of this entire axon. So boom, 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 boom. It's got to happen over and over and over and over and over and over and over again. Okay, so that's going to take some amount of time. It just simply will. Now, when we put on this myelin sheath, what will end up happening is the way that these Schwann cells work, and even the oligodendrocytes work, right? So you have this wrapping, there's a gap, wrapping, gap, wrapping, gap. So you're going to have these gaps, that's what I'm trying to say, between the wrappings. So those gaps, you need to know this term, between the wrappings, between the myelin sheath, they are called the nodes of Ranvier. Okay, so these gaps are called the nodes of Ranvier. So what happens, so if you take a look at this image, so that sodium opening and closing of sodium channels happens here at that node of Ranvier, then basically the signal kind of speeds through this area where you have the myelin wrap, and then the sodium channels open again in the next node of Ranvier. It speeds through the myelinated part, node of Ranvier, speeds through node of Ranvier. So basically it hops, if you will, from node of Ranvier to node of Ranvier to node of Ranvier. And what these images are trying to show you is that, 1 millisecond they all start at the same time, but by 5 milliseconds, look at where this one is versus this one. And by 10 milliseconds, in the myelinated axon, that signal is almost already all the way down to the very end of the axon, whereas in an unmyelinated axon, it's not, right? It's still way up here closer to the cell body, all right? So it's really important that we have the proper myelination, not too much, not too little. You don't want too much because then it chokes off these nodes of Ranvier and you don't have sodium channels opening, but not too little so as you don't have to have this continuous type of conduction. All right? So hopefully that makes sense. The other way that I kind of think about this, just imagine you, okay, and the way you walk, right? So imagine if you had to walk everywhere putting your feet heel to toe, heel to toe, heel to toe. So you were not allowed to skip any part of the floor. You had to walk heel to toe. Okay, it would take you a lot longer than you taking steps where basically your foot goes over a certain span of floor. You walk much faster that way, right? That's the same difference sort of between continuous and saltatory. Continuous is heel to toe. Saltatory, you're taking nice big steps to get wherever it is you have to go. Ooh, so this is one of my favorite things. I wonder, do you guys know what this fish is? So it's a puffer fish, okay? And this puffer fish sort of is on one of these sushi mats, hopefully you can tell. It's actually a delicacy. It's a sushi delicacy, okay? Kind of bring me back here. There's something that this sushi fish, they call it fugu, the puffer fish, has in common with zombies. This is so cool. I just love like zombie connections. Okay, so you're not going to guess this, and you probably already have this word showing up on your screen. What they have in common is something called tetrodotoxin. Okay, so basically what tetrodotoxin is it's blocking the sodium channels. Okay, so it blocks sodium channels. So what ends up happening, a little bit of tetrodotoxin, and it blocks sodium channels. When you put the fugu in your mouth, it kind of causes like a tingling sensation. However, too much, and if we don't have sodium channels opening, you don't have that depolarization. You don't have that depolarization, you don't have any kind of action potential, no signaling going to muscles, right? So people some people die every year of fugu poisoning. What ends up happening is they've ingested way too much tetrodotoxin. You don't open up the sodium channels, and basically you're paralyzed, particularly your diaphragm. Your heart will still keep beating, but your diaphragm won't. So basically you suffocate. You're just like, oh, and you're stiff. Well, you're not stiff, you're actually limp. No muscle contractions. You can't breathe. Now your body will clear the tetrodotoxin after a certain period of time. So people who have this, basically they just put them on a ventilator, keep them, breathing artificially, and after several hours, they're fine. Now, there's a theory out there that the old school zombies, I'm talking like zombies from Haiti, okay? There's a theory out there that where that whole idea of zombies came from is that you had these, I don't know, whatever they're called, the voodoo guys, right? What they would do is they would somehow get to introduce tetrodotoxin into their victims, either via salve or some sort of injection or something. Okay? So these people would become paralyzed to the point where they looked dead. They had very, very shallow breathing that maybe was undetectable. So family members, sad, distraught, it's a tropical area, you quickly buried the dead. Well, obviously these people are going to wake up a few hours later. So the voodoo guy comes along and digs them up and basically says, all right, I just saved you from death. Your family's going to kill you if you just kind of show back up. So you're going to come work for me. And so basically zombie was basically just a slave on a plantation for this voodoo master. And in fact, people do this. This is an issue. So in different cultures, the person's been buried, even if they buried the wrong person or they thought they buried the person and they kind of just, someone, for some reason it's mistaken identity, that person can't just show back up to their family and say, oh, whoa, big mistake, I'm still alive, yay, everyone hugs, The family will want to kill them, but like they think it's a ghost or a demon or something like that coming back. So once these people have been buried and the family thinks they're dead, they're good as dead. There was a case a few years ago, actually, it's probably got to be many years ago now, of this guy, family thought he had died, they had a funeral, and he's like, what? I'm not dead. And he finds out about the funeral, flies back home, shows up at the funeral, and the family tried to kill him because they thought he was a demon. So again, these voodoo guys would take advantage of that sort of cultural taboo, if you will, or that superstition, and basically these people couldn't return back to the villages because they would actually be killed. So they basically, the only place they could live was on the plantation, being slaves for that person. All because of this little tetrodotoxin sort of paralyzing them, and then the family burying them. Kind of a factoid. Hopefully you guys like that. All right, so things that, again, they're contributing to this resting membrane potential. So let's remember, in the extracellular fluid, right, so outside the cell, particularly, you don't necessarily need to worry about the chloride, but really what we're talking about is there's a lot more sodium outside the cell, and then there's a lot more potassium inside the cell, right? And it's going to be the movement of these ions across the cell that's going to cause the depolarization and the repolarization, and therefore the movement, therefore the current down the length of the axon. Anions, remember, are negatively charged ions. So these anions can't leave the cell, so they line up along the inside of that membrane. The positive ones line up on the outside of the membrane. That's how we get our voltage, right? And then remember that sodium-potassium pump is going to reestablish our concentration gradient once the signal has come and gone. And I mean, this is all very, very, very fast. All right. So one of the interesting things, a little bit different than what we saw with muscles, was that, remember muscles, once we had, you had a muscle contraction, if you had another one before the muscle fully relaxed, the second muscle contraction was stronger, then it was stronger and stronger, right? We don't have that in neurons, basically because we can't. Once we hit that positive 30 millivolts, you can't, like the sodium channel's closed, so you can't ever get any higher than that. So you never see a big, you never see it in positive 70 or something. You can't, they're closed. So let's just take a quick look at this graph, what this is trying to say. So the top graph here and this bottom graph line up. So here on the top graph, what we're seeing is the membrane potential in millivolts. So just as we expect, our cell is kind of chugging along at negative 70 millivolts. And then down here in this bottom graph, it's showing the strength of the stimulus. So here in this little purple, magenta colored one, the stimulus is subthreshold. So we see it does cause some depolarization, but not enough to reach threshold, not enough to cause those sodium channels to blow wide open. Okay? So it gets a little bit more positive, in the positive rate, well, more positive, still negatives, but it's a little bit more positive than it was. But then it goes back down, right? The signal does not go down the length of the axon. Now here in this gray, you see that there is a threshold stimulus. So whatever it is comes in, it hits threshold, we have an action potential, right? And then it goes away. Right. Now in this blue, what we see is a suprathreshold stimulus, a really strong stimulus. But what ends up happening is not a stronger membrane potential difference, just more of them. So here's a really big stimulus, and you have boom, fire, fire, fire. That nerve goes, choo, send a signal, 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 signal. So it'll send many, not just one, but many. All right, so hopefully that makes sense to you. So subthreshold stimulus, no action potential. One that hits threshold, one. One that's really much higher than that that threshold, we'll see many action potentials. Okay. Here's a video link on everything that we had talked about the opening and closing of sodium and potassium channels and the myelinated versus unmyelinated axons. I recommend you watch it. Sometimes it's kind of nice to actually see things happen. I think they use a slightly different resting membrane potential number, but just use the numbers that I'm giving you in this class. Different neurons will have slightly different numbers associated with them. Some will have a resting membrane potential of negative 90 millivolts. We're not going to concern ourselves with that. We're just going to use our nice textbook numbers of negative 70 millivolts resting, positive 55 millivolts is our threshold, positive 30 is where sodium channels close and potassium channels open. Okay, those are the numbers that we're going to use in this class. But again, highly recommended. Watch the video. It'll be helpful. All right. So things that are going to affect how fast that signal travels down the length of the axon. Well, the first thing is whether or not it's myelinated, right? We saw myelinated is faster. The actual size of the axon. So the bigger the diameter, the faster the signal will travel. Okay? And then temperature. So the hotter something is, the faster the signal will go, and the colder it is, the slower the signal. Okay? Now, icing, when you've hurt something, icing it will also take down the inflammation, which will take down, having tissue pushing on nerves, causing pain. But one of the other things it does is the signal is so slow, it takes a longer time to actually get to the brain, so you don't register it as so painful. Okay? So it's kind of how you numb the area. Basically, the signal either slows down to such a great degree or it stops altogether. And I'm just going to hide me here. Now, where two neurons come together, we mentioned this before when we talked about muscles, right? We have a gap. So a nerve signal can either flow through that gap, or remember, if the cells are essentially touching, we have what's known as an electrical synapse. So we saw this when we talked about gap junctions, like in the heart and in the intestines. So this allows for very fast communication because all those sodium potassium channels, right, that influx of sodium can go right through that gap junction and continue that action potential in the following nerve cell, right? It allows things to be synchronized against nice synchronized contractions of muscles, like in the heart and then in the intestines. Now, the type of synapse that we're going to talk more about are what are known as chemical synapses. And we saw this with muscle cells, right? We saw that the length of the axon went down to the muscle, to that motor end plate, and then there was this tiny little gap between the two, right? And then that synaptic cleft, right, that tiny gap between the nerve and then the muscle. And we actually had to use neurotransmitters, acetylcholine, to get across that gap, right? So basically that's what all the other neurons in your body really do. So let's take a look at this image here. Right, so here's a single synaptic end bulb. Right, so here's what we know as the presynaptic neuron. So I just want you guys to get comfortable with that term. So in this case, we're talking about two different neurons. We're not talking about neuron and muscle. Okay, so here's a presynaptic neuron. Here's that synaptic cleft, and then that means that this is simply the postsynaptic neuron. All right. So again, it's just saying that this neuron is before the synaptic cleft, this one is after. All right, again, we're talking about neurons. So in this axon, or in this neuron, I should say, in the presynaptic neuron, we have an action potential. It's coming down that length of the axon, and here we are in that synaptic end bulb, which is basically the last bit of it. Okay? As that nerve signal, all these sodium channels are opening and closing, when we get down to the synaptic end bulb, what's going to happen is that those sodium channels, the depolarization, is actually going to cause calcium channels to open. Okay? So at that synaptic end bulb, the depolarization is going to cause calcium channels to open. So calcium is going to come into the cell as you see in this image. Okay? Again, these are voltage-gated calcium channels. As that membrane potential changes, as this nerve impulse comes down the length of the synaptic end bulb, it causes these calcium channels to open. Calcium comes into the cell. Now, why that is so important is it's basically this calcium coming into the cell that will stimulate these synaptic vesicles to bind with the cell membrane of the synaptic end bulb. Okay, so again, calcium coming into the cell, it's this calcium that will cause these synaptic vesicles to bind with the cell membrane and then therefore release their neurotransmitters into the synaptic cleft. All right, so hopefully that makes sense to you. I'll just say it one more time. Nerve signal comes down the presynaptic neuron, and by that, all that sodium, potassium channel stuff that we just talked about. When it gets down to the end bulb here, it causes the opening of voltage-gated calcium channels. Calcium comes into the cell. That stimulates these synaptic vesicles to bind with the cell membrane, and once it does that, it causes the neurotransmitter to be released. Now that can be dopamine, that can be serotonin, that could be a whole bunch of different ones. We're not specifying right now, but that's basically how it functions. And once that neurotransmitter is released, it goes across that synaptic cleft. It binds to the receptor, right, on presumably the dendrite of another nerve cell of that postsynaptic neuron. Okay, and then once it binds, these ion channels will open, and the nerve impulse continues. All right. So hopefully that makes sense. So again, it's to release these neurotransmitters across the synaptic cleft. It binds to receptors, causing them to open. And then as you can see, sodium continues into this cell. That causes a depolarization in the postsynaptic neuron, which can cause a nerve impulse. And then the cell signal is transmitted to another part of the brain, another neuron, to another neuron, to another neuron. So finally you go, aha! That person is Liz. She has a black shirt on. You know, whatever that signal is that you're interpreting. Right? It allows you to bring up people's faces, you know, their names, associated color, so you know, whatever the larger processing is. Okay. Hopefully that makes sense to you. Please do not hesitate to email me. Let me know if you have any questions. I understand these are more complex topics, so I will try to clarify as best as I can. All right? All right, stop the recording here, and I will see you in the next recording.